Hello, everyone. Thank you so much for joining our live stream today. My name is Erin Spencer, and I am the communications manager for golf restoration at the National Marine Sanctuary Foundation. And I'm going to be your host today for this program where we're going to take a closer look into what our restoration teams are doing at sea. Soon we're going to go straight to a vessel that's currently in port with a lot of our restoration team. But first, I'm going to do a little bit of an introduction to this work and give you some background. Throughout today's program, please feel free to share any questions that you have in the chat, and we will make sure to address those and ask our team on board. Just toss them in anytime. We will answer them as we go. Also, we would love to get your feedback on today's program. There is a short survey that you can fill out after the program. We'd love to hear about your experience and any recommendations that you might have for us moving forward. We're going to put the link in there for now, but then at the end of the show, we'll put it there again so you can revisit after the show. All right, so in just a few minutes, we're going to connect with the folks on board. But first, let's look a little bit closer into the background of this restoration work. As you travel deeper into the ocean, less and less sunlight is able to penetrate the water. Just past recreational scuba diving limits, you reach the mesophotic zone, which means middle light. Despite the low levels of sunlight, this zone is home to colorful corals, sponges, fish, and other invertebrates. And then beyond the twilight zone, there is no sunlight at all, and it gets much colder and there's higher pressures. But despite this, there are still vibrant habitats full of life at these depths, hundreds of thousands of feet below the surface. These mesophotic and deep benthic or seafloor ecosystems are found worldwide, including in the Gulf. However, the 2010 Deepwater Horizon oil spill dealt a heavy blow to Gulf communities on land and at sea. 11 people lost their lives, and natural resources, wildlife, and habitats across the Northern Gulf were severely impacted. More than 770 square miles of deep sea habitat were impacted by the spill. But as a result, collaborative restoration projects are currently underway to help us understand and restore these seafloor ecosystems. The projects are developing new and improved maps of the seafloor to locate these important coral communities, learn more about the species that live in these habitats, the environmental conditions they thrive in, and how they might be connected across the Gulf. Determining the best methods to encourage deep sea coral growth and reproduction to help restore impacted coral species and the important habitats that they create. Also reducing the threats that these ecosystems face, like marine debris and other threats on the seafloor. And then also connecting the habitats and the restoration work to the public and natural resource managers through programs like this one we have today. This restoration work is made possible by the Deepwater Horizon Natural Resource Damage Assessment Open Ocean Trustees, and it's led by the National Oceanic and Atmospheric Administration and the Department of Interior, alongside many partners, including a large number of academic institutions and non-governmental organizations. And we have some opportunities to learn more about this work here, and then we'll paste some more links in the chat throughout the program. So now it's time, I think, to jump to our friends on the RV Point Sur, which is a 135-foot vessel owned by the University of Southern Mississippi and is operated by the Louisiana University's Marine Consortium, which is also known as LUMCON. Today, the team is back at the port of Gulfport, which is their home port. They came back a day early because of some weather conditions, but it's fortunate for us because now we get to see this live footage of the vessel from the outside, which is something that we really don't get to see. So this is coming to you live from Gulfport. We have one of our team members showing off the exterior of the vessel. This is a vessel that our team works with quite a lot. It's really well equipped to tackle a lot of the different restoration objectives that we have. And you can see just as we're walking on board the vessel, you see a great view of the ROV or remotely operated vehicle that was used on this expedition, Global Explorer. And we're going to see some more footage from that ROV. It's a really, really important tool in our restoration work. So here, as we're walking on, you see the back deck of the vessel with Right now they're in demobilization time, so folks are packing things up, getting ready for the next leg of the expedition. But there is typically when you're at sea, everyone is equipped with their hard hats and their steel-toed boots and ready to rock. As you can see, there's a lot of heavy machinery on the back deck there. Great view of the hard hats there as well. And you can see it's a great opportunity to outfit your hard hat with stickers and all sorts of other cool things. So now we walk into the vessel inside. This is the air conditioning, very important part. And now we are live in the lab, ready to talk to our team. Hi, team.
Hello.
Hey. Thank you so much for joining us today. I'm sorry to hear that you guys had to come back in early, but we're really excited that you are able to chat with us now. Let's go ahead and get started with some introductions. Would you mind going around and telling us your name, who you work with, and what your role is on this expedition?
Yeah, so hello, everyone. I'm Jack Gerard. I'm with the University of Rhode Island, working in the Prada lab. We do, I guess, a lot of fragmentation of octocorals. So we're working on settlement of octocorals and a lot of, I guess, marine genomics and eDNA. And I'll pass it off to Brittany.
Hi, everyone. I'm Brittany. I'm the lab manager at the Prada lab at URI. My job is a lot of the actual experiments that take place, to get the DNA out of the corals, and we also work with environmental DNA.
Hello, everyone. My name is Haley Alexic. I'm a research assistant in the Prada lab at the University of Rhode Island. I kind of just help out with the little things in the lab. I do whatever they need me to do. Just, I don't know, help out in the lab, basically.
Hello, my name's Lizzie Welch. I'm also a research assistant from the Prada lab. I do kind of the same stuff that Haley does. They give me a task, I complete it. I do help them a lot with getting projects done, doing DNA extractions, etc. So it's fun work.
Well, thanks for the introductions. And I think you touched on something that's really important that'll be a theme we keep coming back to, which is when you're out at sea, oftentimes you wear a lot of different hats. And it truly is a all hands on deck situation, especially when some of these pieces of equipment are coming up from the seafloor. So I want to kick things off because, of course, this program today, we're talking about tiny creatures. And I think a lot of people, when we think about the deep sea, we think about things like giant squid or whales that dive deep. But we focus on a lot of things that are much, much smaller than that. And we're going to learn today some of the ways that we use to study some of these tiny creatures. But to kick us off, when we talk about tiny creatures in the deep benthic zone and mesophotic zone of the Gulf, what are some of the animals that we're talking about here?
Yeah, so I think a good place to start is what's directly growing on the bottom and things maybe you would see first, right? So you'll find a lot of those flexible, soft corals, depending on where you are. You might find some hard corals. And now, I guess what I would challenge you to do is take the high level view and think of a forest and then keep zooming in, right? As you zoom in, you're going to start finding birds and squirrels. And right, if you were to then get at the level of where grass and dirt is, you start now looking at bugs. And there are basically marine versions of these things. You'll find very, very small fish. You will find shrimp, squat lobsters, nudibranchs, various forms of algae. It gets quite extensive and mind-bogglingly diverse, but that's what we're looking at, is essentially going out into your lawn in your backyard and pulling back the little blades of grass and seeing what crawls around in there. Anybody got anything to?
That was really good.
That's a good.
That was awesome. No, that was a perfect coverage. And why are some of these smaller animals really important parts of this Gulf ecosystem?
Yeah, so right, you think of your giant squid or your sharks and all these things, and they play their own roles, but they have to eat, right? They have to live in an environment, and a lot of those will say either bottom-up controls or even simply the food is coming from what's coming off of the bottom, right? Everything's playing its own role, whether it's decomposing something, eating something else, and you build an ecosystem out of those things. It's very hard to build an entire ecosystem out of a squid or a whale. So it supports everything else would be my answer to that.
Small, but very important and very mighty.
Yes.
I want to get into a little bit deeper about how we are able to study these small small creatures in the Gulf, partially because already these mesophotic and deep benthic habitats are hard to get to. It's not as easy as tossing on a snorkel and looking directly at these animals. And then even further, now we have to look at that super fine scale of some of these smaller animals. So I know that all of you here are working on different methods. And so I want to start first talking about some of these settlement arrays. And I know, Jack, you were spearheading some of these settlement arrays. Can you tell us what a settlement array is and how you've been using them on this expedition?
Yeah, so our settlement arrays are asking a couple of very specific questions centered around how are corals recruiting, be that how much, how often, and then starting to get into some of the variety of different sites and different time periods. The major restoration application for this is that if you're going to scale this up and start putting larger structures down, if there is a settlement choice, you want to know how the corals make those choices, or at least if you can't get the how, you want to know that they're making certain choices, choosing certain things over others. So as you see, we have the settlement arrays. You can tell there are different surfaces. We've made them out of different materials. We have put them in different orientations, right? Some of these things are very simple, right? Do they want to be on something vertical, on something horizontal? Does it matter if there's more light, if there's less light? And they're basic questions, but they're very important questions. And it's very hard to do it in the mesophotic because, as you say, you can't just go out in your snorkel, kick down, and set these things up. So there ends up being a lot of planning between how are we going to go down there? How often are we going to come back? What vehicles do we need to take these photos? And so the arrays are set up to basically answer that kind of question. But because there's no wasted space in the ocean, right, like we can't choose what settles there, which is part of the point. You'll find bryozoans all over it. You'll find Cariciterious algae all over it. So in that sense, right, we're going to be taking lots of photos, and for other people or other scientists that want to use that information, it kind of is just this treasure trove of ways to ask different questions or look at just how this environment works. Oh my goodness, there I am making up.
So essentially, you're trying a lot of other animals, like the little habitat that you're putting down there as well. And Brittany, I know that you worked on these as well. Can you tell us a little bit about the process of how you deploy and recover these settlement arrays?
Yeah, so the locations of the arrays are based on whether we want them on reef or on sand. These big arrays get put together, and then we send the ROV down to pick a good spot. And then we lower them down and they stay there for a year or 2 or sometimes 5 years. And then it's pretty much the same thing coming back up. We use the ROV to find the arrays, we hook them on, and then we pull them up, and then we process them all very quickly in a short amount of time.
And I think what we're looking at here is some footage of the deployment of these settlement arrays from this mission that you're on right now. Is that right?
Yep, it is.
That is so wild to see. From mixing cement, was that cement, Jack, that you were mixing up?
Yep.
All the way down to the seafloor.
Yes, yeah, that was cement. And some of the beauty of this project is you can go into Home Depot and just, take an afternoon or two, kind of pull it all together. So not super high tech, but gets the job done.
Do you ever explain to the folks working at Home Depot when they ask if you need help with anything, like what you're working on?
Yeah, and sometimes I'm proud when I can stump them. Usually they get really curious. The limestone tiles, although I don't make them, when I told the company that that's what I was doing, I had this whole email exchange with this woman that was like, "Oh, my mother's a marine biologist. What are you trying to count or study?" And I actually was at a hardware store trying to make the bulkheads for the coolers that we keep them in. And oh my goodness, those guys got fascinated. I was just there the other day showing them pictures of how well it all went. But like people kind of light up, like especially hardware people. They're like, "Oh yeah, let me try to solve this problem for you." Probably because it's so different from all the other stuff they're usually trying to work on.
I absolutely love that. And also goes to show that like outreach about this work takes all kinds of forms, whether or not you're trying to troubleshoot building something that'll end up on the seafloor, and people want to participate and help.
Yes.
I want to talk a little bit more about what actually happens when these sediment tiles are processed. So Brittany, I know that you worked some with eDNA, and can you talk a little bit about that and include why we reference them as smoothies sometimes that you're working with?
Yes. So when we bring up the arrays, we essentially take all the tiles off. We take pictures of everything. We take our little samples, and then we scrape everything that's on those tiles into a slurry, I guess. And we put it in a blender, and it looks like a very suspicious smoothie. But then we, take that sort of blended material, and we can extract DNA from it. So eDNA is really cool because it's a non-invasive sampling method. So for cryptic species or things that are threatened or endangered, we're not actually disturbing them or their environment. We are just taking DNA that's, easy to get to, usually water or sediment when it comes to marine biology.
This is awesome. And it is all happening in that room that you're in right now. And I imagine it gets pretty crowded as you guys are all trying to do this at the same time.
Yes, absolutely. It does get crowded. Yeah, it's a lot of work in a very short amount of time. And there's, everybody's working together and there's a lot of collaboration, but that does make for close quarters.
Awesome. Well, this is such great footage of the settlement arrays. I want to jump into sediment tiles. I want to jump into some other methods that we were using on this expedition, because this was just one of many things that you all were working on. So Lizzie and Haley, I know you're on board. You were working with these settlement tiles, but you were also working with something called arms. Can you tell us about what those are and how you were involved in the process of the arms?
You or me? Okay. So the arms project, we were working with some of the scientists on boards, and they are kind of similar to the arrays that we were working on for Jack's project, but a lot of them are stacked. I think there was 9 plates on each structure. There it is. There's 9 plates stacked on top of each other with space in between, all on like this large base plate. And it's similar to what Jack's project. It allows for more space of things to settle. So it provides open area for benthic organisms to settle on them because space in the ocean is so limited. And we would process them in a very similar way as we did with Jack's. We'd take apart all the different plates and set them all aside and then see what settled on what plate, pick out little species, see what settled where, and then we'd also do eDNA samples for those as well, trying to figure out what exactly is settling and just to get a general survey of ocean populations in benthic environments.
This is awesome and great footage of what these look like on the seafloor. What are some of the most unexpected or interesting things that you've seen on those arms, or I guess the settlement tiles?
On both of them, we got like some of my favorite benthic like little organisms are nudibranchs. They're little sea slugs. There's a ton of varieties of them. We got a lot of little nudibranchs, and we got a specific one called a sea bunny, which are really, really cute. They have like a little pom-pom on their tail, and they have little antennas. They're really adorable. And we got a bunch of things like we got some brittle stars, we got some crabs, there were snails, there was all types of different groups of organisms just in these little areas of plates. So I think we got sea urchins as well. I think the sea stars and the crabs and the nudibranchs are some of my favorites.
Yeah, I agree. I kind of like all the small little sessile and motile invertebrates. I think they're all super cool, and it's really cool how you can look at their morphology and tell like what they're kind of meant to do in their ecosystems. I really loved the squat lobsters because of the way they move. Like their little fin like goes like that, and then they like go backwards. And I thought that was really cool. Is that? No, I think that's a crab. Yeah, but the little crabs are super cute as well. Yeah. So
look at that guy.
Yeah, and that's arrowhead crab.
Arrowhead crab.
So yeah, we picked off all the creatures from the settlement arrays that like looked interesting, and then we had the Smithsonian team like process them and take them in. But yeah.
And if you're watching right now and you want to learn more about arms and how they work, we actually had a whole live stream that aired yesterday that is available for viewing after the fact. And we'll paste that link in the chat. And it's a great way to dive a little bit deeper into some of the specific methods about arms and how they were used on this expedition. But I'm also thinking maybe can we get a up close look at a processed sediment tile, or is that something that you have here in the lab?
Yeah, absolutely.
We kept some just for you all.
Just for us. Oh, that's fantastic. Okay, can you walk us through what we're looking at here?
Yeah, so this one's been completely processed. So it's gone through the scraping and we let it dry overnight. So this one is one of the concrete ones. All the little rubble on there is basically the aragonite reef rubble we've added to give it some complexity and allow things to settle in. This would be, I guess, the bottom side. Again, we've processed this. So we've pretty much scraped it clean. I don't know how well it's coming through. You can tell there's still maybe little bits of dried algae or a couple small tubes on there. But if you want, we kept 2 of them. We have them in some bins. We did not scrape them. And you can tell they'll look a lot different than these with all the stuff on them. So are we good to
would be awesome.
go over to that?
Yeah, let's go see that.
Sweet.
It is cool to see that it definitely looks different from when they were deployed. Looks like they've been through a little bit of stuff in the deep sea.
Yeah, the cooler's in the back. I thought that was gonna be a lot heavier.
That one was heavy. It's full. Anyway, so here we have basically our 2 examples. We have our concrete tile and our limestone tile, and you can see, visually they do look quite different. The, I guess, job of the science here is to figure out 1, what is on there, and then 2, why are they a little different? Is it because of the complexity? Is it because of the material? Is it because they were set at certain angles, be it horizontal or vertical? So that's going to be my job for probably the next 11 months of the year, is figuring that out. All right, so here's our limestone tile. This would be the top side.
Oh, wow.
And we have the back side. Right. I don't know if you can get the depth there at all, but you can see there's little guy, like those little stocky things might be bryozoans. We got a little worm, little casing there. Again, I'm not sure how well the focusing is working.
No, we can see it. It looks great. But they're definitely little guys on there.
Right. And then the concrete tiles are always fun, right? You can see there's just so much growing on there. I think we got this little red thing. You have some tubes there, a bunch of hydroids over here. Maybe I can't see if there's a holdfast or not, but every now and then we find a black coral on one of these things. But what I do always find really cool is how much you can tell, like, light is really important. So that bright streak moving through the center of the tile is bright because that's where the zip tie was for a year. And you can, right, it doesn't get any light. So it tells you that all the other dark stuff growing around it, right, is probably photosynthetic, or there's some type of algae on it. When you look at the back side, you see how bright that is? Right, that was the dark side. When we put them in, they were both maybe not. Yeah, this was probably a little darker than when it went in, but the difference between them to me is really striking. And it just shows you that the organisms respond very well to their environment and where they choose to be.
That's so interesting how you can tell so much just by looking at it as is when it comes up. And then, of course, as you said, it'll take a long time to dig into all of those findings from each one of those tiles. That is awesome. Thank you so much for showing that to us. I also think that a lot of people have questions, not just about some of the science that you're doing here, but also what it is like being on board. And I understand this was a particularly interesting expedition that you encountered some conditions while you were out at sea. And I know for some of you, it's your first time on one of the restoration expeditions. And I'm wondering if you could share a little bit about your experience for your first time at sea and what it was like.
Yeah, so it was mine and Lizzie's first time on a research cruise, and I personally had an amazing time. One of the challenges, I think, was like the boat rocking, like actually being like, I'm used to being in a lab and working in a lab, but when it's moving, it's like a whole new game.
Yeah.
Because your samples are moving around, you have to get the sticky pads to keep your samples in one place, and then if you're focusing on one thing for too long, you get seasick, and then you have to take a break. So yeah, that was like the main difference, I guess. But it was really nice how everybody was collaborating. Like we had our team, the Prada Lab, and the Smithsonian team And when we were helping with their stuff, they were helping with our stuff. Like everyone kind of was doing similar things, and it was just a really collaborative environment, which I enjoyed.
Going off of that, like there was the first day was some of the roughest weather I think we had all week, so everyone was struggling a little bit to keep themselves together. But once everyone got their sea legs, it was smooth sailing mostly. Everyone was able to get some work done by the end of the day, and I think one of the best parts about like being on a vessel like this is when you need a break, you can just go outside, watch the water pass by. A lot of times we had a ton of like sea creatures following the boat, or they'd be things jumping out of the water, there'd be birds flying with us. So I think that's one of the most rewarding parts, other than getting the work done, is being able to just like take a break, enjoy the environment that you're in, and realize that like you're out on a boat in the middle of nowhere and you're doing a lot of things. It didn't feel like I was working for much of the cruise because it was so much fun. The pictures here, we got the Atlantic spotted dolphins were following the boat for about 2 days of the cruise, and it was really, really cool to see them like feed and interact with each other. So that was one of the super neat things about being out here.
And I know that we're talking about little small creatures here, but I think everyone loves a good megafauna video, right? While you're out at sea.
Oh, yeah.
Oh yeah, that's fantastic. That's fantastic.
We got to see all walks of life from the ocean, big and small. It's very fun.
And I think it also, that video earlier showing how the seas were rocking, it really puts it into context when you think about how closely you have to look at those settlement tiles that we just saw, right, as you're holding it up, and all these small animals. And the idea of having to focus on that in the lab setting while also trying to keep your balance is very impressive. So congrats to you all for being able to get so much done on this expedition. We're getting a little bit close to the end of our time here. Just a reminder that you can add questions in the chat. I'm going to look really quickly and see. Oh, we do have a question. We have a question. Did you encounter any benthic ctenophores? Sounds like someone that has a little background on ctenophores asking this question.
I didn't see one. I feel like Mark would have pointed it out if we saw one. Ctenophores are like these. If you've never seen one, they're like jellyfish, or they're called comb jellies, and they have these tiny little things called teens. And generally when you find them, they're beautiful because they beat them, and there's rows of them, and they catch the light and make these little rainbows.
Yeah, they blink, and it's really, really cool. I don't think we saw any though.
Yeah,
so unfortunately,
there were some like gelatinous creatures on the tiles.
Okay,
I forgot what they were called. Something with an A. I don't remember the scientific name, but there were some gelatinous creatures that were cool.
Yeah,
I didn't see any personally. That doesn't mean that they weren't there. The Smithsonian was all over just pulling these things apart. So it's a good question.
And I think that also brings up a good point that you've touched on a couple of times, which is there's so much that happens while you're actually at sea, but most of the work and most of the data and the findings really comes in the months after, where you're analyzing all of these samples and looking at all of the data. And that's where those genetic tools, for example, are really important in figuring out what was actually there. But I also have to imagine that as you're pulling those sediment tiles or those arms out, it is kind of like a bunch of scientists in a candy store. You could just feel the excitement of it. Yeah, because you never know what you're
being in a
see.
candy store for 17 hours a day.
Yeah. And then if one of us doesn't know what a species is, we'll just yell out and call somebody else who might know what it is, and then they come over and they ID it. And
it's a lot of back and forth, but it was nice to have them around.
Yeah.
I truly think there's nothing like more exciting and energizing than watching someone get super excited and nerd out about an animal that they found on one of these here. And this was actually from our live stream yesterday, and you can see there's some really, really fine scale photography that they do of these arms, and the photography is a really important tool as well in identifying these. So don't forget to check out that one from yesterday if you're interested in learning more about the arms. So as we are approaching the end here, I want to circle back and really tie all of this in to, can you give us kind of a summary of how all of this work that you're doing right now really helps contribute to restoration of these ecosystems?
Yeah, so when it comes to restoration, you have a couple options. One option is active, another option is passive. Passive usually means you're going to keep people out, let the space recover on its own. But in order for you to make that decision, you need to know, one, that it will, and two, at what time scale would it come back. So if we're using corals sort of as a proxy for, right, when we fly over with an ROV and we see a lot of these large octocorals, and call it an octocoral forest or a meadow or whatever your analogy is, right? It's kind of, again, like asking the question, when we log a forest, how many years do you have to wait before that forest regenerates? What we kind of don't know or are trying to figure out is, how long does that take? And one of the questions to figure that out, like trees, right, the seeds are going to move and fall somewhere. The coral propagules, the coral planula, the larvae are going to settle somewhere at some rate. And the settlement tiles basically get at that question for the space that they're put in, while also trying to address, are there preferences? The preferences come into some of the active restoration, which, all right, if we're going to put out substrate and this place can recover on its own, but we don't necessarily have to cut corals and fragment and move them around, we'll place the substrates out, we'll try to facilitate settlement, facilitate growth, and just let it play out naturally. The other option, right, so say we don't find a lot of coral settlers, or it seems that it's going to take decades to centuries for this to recover on its own, then you come down more of an active management path where you start doing coral fragmentation. So you'll find, all right, are there some corals that are there? Are they healthy? Can we take clippings of them, spread them out, and allow them to grow so that they all sort of grow together? And depending on the severity of the impact of any one site, you start then asking the question, are there healthy populations other places where we can take fragments to put them into a place that used to have corals that no longer does? And a lot of, right, the settlement sort of gets into a facet of that, but these are the questions that you have to ask, because you don't want to be fragmenting corals from one place, putting them in another if you don't have to. And when you start doing the active fragmentation, you also want to know that when you take them from somewhere, they have a good chance of surviving somewhere else. So you have to do a lot of work on the back end to understand, how far can you transport corals? Does it matter if you take them from 10 meters shallower and place them 10 meters deeper, right? You got to be very targeted and very methodical. So that's, the work ahead, and that's the work that we're doing as we try to scale up and figure out, how are we going to do it?
I think that is such a great overview. Thank you. And also, I think really speaks to how interconnected all of these restoration questions that you're addressing are, and how they all work together to really help contribute to this broader recovery of this really, really important Gulf habitat. So with that, I think we will wrap up for today and let you guys get back to breaking down the lab and wrapping up the expedition. But just want to say thank you so much for joining us. Really, really appreciate you taking the time, and loved getting to see what was in the lab, and really wish you the best of luck on all the analysis that is to come on your samples. After
Thank
Thank
you
you very much.
Thank you.
Thank you. And safe travels back.
Thanks.
Thank you.
And safe travels to them. And for everyone tuning in, thank you so much for taking the time to join us, and thank you for your questions. If you liked today's event, we have a lot of other live streams in our library, and we even have some coming up. The next one is next week, I believe on October 14th at 2:00 Eastern, and we're going to be looking at coral imaging. So please mark your calendars and join us then. We're going to paste in the chat. You can find some past live streams, and then also see all of our upcoming ones in the link from the University of Rhode Island's Inner Space Center YouTube page. You can also learn more about these projects, find articles, publications, and we have this really wonderful interactive story map that you can see. You can check for links for that in the chat as well. And also, as a reminder, we would love to get your feedback on today's program and let us know what you enjoyed, what we can improve, and what you'd like to hear about on future expeditions. So thank you again so much for joining us, and we will see you next time.